Hi everyone, I'm Adrian Harrier. I'm a researcher for IGL. In terms of my studies and expertise, I have a undergraduate degree, bachelor's degree in biomedical sciences. I also have a master's in molecular and cell biology. My experience and knowledge mainly lies in the applications of biomedical techniques such as PCR, electrophoresis, and many more in the disease diagnosis and pathological studies. So for today, we'll be talking about bacteria. So bacteria can be found nearly everywhere. They have adapted to be able to survive in extreme conditions as far as even the Earth's crust. There are currently around 30,000 named species of bacteria. They come in a range of shapes and forms from spheres, rods to spirals. Bacteria were actually among the first life forms that developed on Earth. They helped shape our world in more ways than we can ever imagine. When most people think about bacteria, they immediately tend to think about the nasty diseases that they can bring to us. What not a lot of people know is that it is more than just a pathogen. In fact, a lot of bacteria are actually not that harmful, and they have characteristics that make them suitable for a number of positive applications. Some of these applications can be used for fermentation, for example, to make foods such as cheese, wine, yogurt. Others are used to decompose sewage, while others are also used for the production of antibiotics and vaccines and other medical treatments. Certain biological characteristics of bacteria have made it very useful for us for a very long time. Modern science is currently harnessing and making use of these characteristics to meet our own needs. The beginning of the history of bacteria began shortly after the beginning of the Earth itself, around 4.5 billion years ago. Some fossil records show that bacteria first appeared 3.7 billion years ago. All life on Earth began with these single-celled organisms. As time went on, the tree of life diverged to produce two main categories, which were the eukaryotes and prokaryotes. The difference between the two is the structure of their cells. Eukaryotes are typically multicellular, larger, and have more complicated cells. These can be organisms such as plants, fungi, and animals. Inside these cells, they have a nucleus, as well as other organelles that are enclosed in membranes. Prokaryotes, on the other hand, consist of just one small single cell. Their organelles are not enclosed in a membrane. Bacteria falls into this group. The number of bacteria on Earth is estimated to be around 5 million trillion, or 50 with 30 zeros. This gives rise to one of bacteria's superpowers, their ability to rapidly evolve and adapt to their environment and food source. In nature, bacteria often grow in colonies. Populations of these colonies can vary from 100 million to 10 billion. Bacteria can also have short generation time. This is the time it takes for bacteria population to double. This can happen in as little as 20 minutes to 10 hours. With its biological characteristics, they have the ability to adapt and survive in different kinds of environment. For example, when bacteria is exposed to an environment with a new food source, they just evolve to eat it. And also, scientists found that 350 to 450 million years ago, there was a group of bacteria that acquired a gene that allowed them to break down chitin. This is a fibrous material that was found in the exoskeleton of arthropods. They were very abundant at that time. However, when utilized in the right way, these high metabolic properties can be useful in breaking down things that we normally cannot. Like all living things, bacteria need nutrients to survive, and more specifically, they need fixed carbons, or carbons that are incorporated into organic molecules. It's what gives us energy to move, grow, and reproduce. But it's not as simple as eating and getting energy. Inside our cells, a series of chemical reactions take place that break down nutrients that our bodies need to grow and thrive. This is known as metabolism. While we can only eat organic materials such as plants and animals, bacteria can create their own fixed carbons from light. They can also carbonize inorganic molecules like carbon dioxide. Having these options make bacteria more metabolically diverse than us eukaryotes, meaning they have a lot more options when it comes to nutrients. In fact, scientists have found bacteria eating complex compounds that are harmful to the environment. Several strains of bacteria even have the ability to digest hydrocarbons, like those found in oil. After a massive oil spill back in 2010 in the Gulf of Mexico, resident bacteria digested 200 tons of the oil and natural gas, aiding to the cleanup efforts by converting these harmful substances into CO2 and water. Oil is just the beginning of the environmental cleanup abilities of bacteria. In 2016, a group of scientists in Japan found a colony of bacteria that was able to digest PET. This is a type of plastic that is commonly used for making water bottles. Each year, we produce around 33 million tons of PET, and they are very difficult to decompose compared to other types of plastics. When grown on PET, this bacteria produce two key enzymes. The first breaks down PET into MHET, then the second enzyme breaks down the MHET into the two main components that make up PET, ethylene glycol and terephthalic acid. The bacteria can then use these as carbon source. However, this process takes time. It takes weeks for the bacteria to degrade 60 milligrams of PET. However, in recent years, scientists succeeded in making a slight change to the structure of the enzyme that increased the degradation rate of PET by 20%. Now, only taking a few days to degrade the same amount.